Okay, so my task will be in the next couple of minutes to bring these quantitative geneticists together with this guy in the background, so which all of you know, I guess. Yeah. So the very prerequisite for plant breeding to work is, of course, genetic variation. And if we look at different traits, well, we see different patterns of variation there. For example, Mendelian traits, they really show discrete states. For example, you see aunt versus non-aunt, wheat heads here. On the other side, of course, we also have, let's say, more quantitative traits that show that follow a continuous distribution. For example, here in maize, you can see very nicely a lot of variation, different ear lengths, colors, kernel sizes and shapes and so on. And sometimes, that's we see a combination of these. It means a mixture of discrete and continuous variation. For example, infected versus non-infected with a pathogen versus partially infected. It means, for example, here it's an example of a parasitic fungus in wheat, which is called stripe rust or leaf yellow rust. You can clearly see here, it's infected versus non-infected. If you just look at two genotypes, and if we really test a lot of genotypes in the field, you will see this kind of continuous variation, where if you then really have a close look at the leaves, some genotypes are completely resistant, then moderate resistant, or completely susceptible. So, and then question is, of course, what's behind this pattern, and how can we breed for this? And basically, if you just look then at a lot of genotypes and look at this distribution, you would see that some would fall in this class, which of the completely resistant genotypes, and some show this kind of continuous distribution. Well, first explanation is basically given by Mendelian genetics. And to have, so to say, a look into these patterns, one would conduct a very classical experiment, quite similar to what Mendel has done. But here we just cross, so to say, a susceptible and a resistant parent, which, and let's say there's just one gene behind this resistance, this example. Cross them and look at the F1 generation, and based on the law of dominance and uniformity, they would be all resistant. And if we get progeny from such a cross, you would see the classical segregation pattern here, what also Brigitte just showed. Just in this case, then you have some, at least, 1/4 would be carried would be homozygous for this resistant gene, and 1/4 would be so to say, homozygous for this, let's say, susceptible gene, or would not carry this dominant resistance gene, and then 1/2 would be homozygous. And if you really look then in the field, you would see this classical 3-to-1 pattern in the field. So to say, 3/4 would be resistant. So, and what means, so to say, susceptible or resistant here? Susceptible would just mean, let's say, you have a fungus, and then the fungal spore lands on the plant leaf, and then it just penetrates, so to say, the plant leaf, and then grows inside, infects a lot of cells. In the case of, let's say, such a parasitic fungus, it will just kind of not kill the host or the plant immediately, but just leach nutrients from it. And of course, these nutrients would then not be available to produce kernels, and the yield will be strongly reduced. On the other hand, resistant can mean in this case, so quite often a so-called hypersensitive reaction. It means the fungus grows, and then maybe it starts or tries to infect one cell, but the plant can recognize this, and then, so to say, this cell self-destructs, and fungus cannot grow anymore, and the plants are resistant. And in such a system, which follows very classical Mendel's rules, we cannot only look at the genotype of these progeny or the plants, but we also have to consider the genotype of the pathogen, because for every gene of resistance, there is a gene for, let's say, susceptibility in the pathogen population. And this gene for gene hypothesis has been already described in the 1920s by Flohr, or 1930s, but it's still one of the most important concepts in plant breeding up to date. And it means just if this plant is just resistant, if this resistant gene comes together with the according gene for avirulence, this large A gene from the pathogen, and then quite often they show this kind of hypersensitive reaction. Plants are completely resistant. It means this basically can explain kind of the part of this pattern, so to say. It means here we can just now say, okay, the plants are completely resistant because probably there's one of these major resistance genes, and yes, and if they genotypes don't carry it, then they show some kind of disease severity, but there's still some distribution can be seen here. So, but this does not explain why we have still this kind of distribution of these susceptible lines or genotypes. So, and at the same time, more or less like Mendel, there was a second school, not this school of biometrical genetics, and this Francis Galton was one of the most important practitioners there. And they looked more into these kind of distributions of populations, for example, by just looking at the means or the variances. And they introduced a very important concept of heritability. And let's say we just looked then at the mid-parent value, so to say, of the father and mother, and then we look at the progeny and plot them against each other, and using regression-based methods, one could then just show that the slope of this regression curve you see here, that's called the heritability. And with this heritability, this kind of is a measurement which can then be used to predict, so to say, if we cross now this parent A and parent B, will the progeny be more resistant or more susceptible? Because based on the heritability, we would assume that if we cross now two more resistant parents among each other, then also the progeny would be more resistant, which is kind of logical, I guess. And of course, this explains then a bit, if we go back to this distribution, we see more, more the general pattern here we see. But not really what's behind it, so to say, genetically, like we have seen with this Mendelian inheritance beforehand. And so a smart guy, Ronald Fisher, he set up to a task to unite these two schools of genetics in the 1920s also. And he came up with a, let's say, pretty simple method to do this, or model. Just let's assume you have now a gene is controlled by a single gene, trait is controlled by a single gene with two alleles, a large P and small p. And then you have egg cells and sperm cells, and then if they come together, then we will see these genotypes which carry, so to say, twice this large P allele. Others are heterozygous, so small p and large p, and others will have two small p's. And now, if we assign, let's say, phenotypic values to each of these alleles, let's say p equals 50, and then we sum this up, then you will see, for example, this small p will get a value of 100, and this small p large p and this small p large p, both of them, you can see here, they will get a value of 50, and they occur with a frequency of 1/2. If one just plots this, one could already see, okay, you can see this kind of distribution here. And now you can just continue with this exercise, so to say, and say now we don't have one gene, but two genes, with alleles large p, small p, and large q and small q. And yeah, if you cross them among each other and look at the progeny, you can already see 16 different genotypes. And then just repeat this exercise and assign phenotypic values to each of these alleles. Now, let's say 25. And then if we then plot this again and how often each phenotypic value occurs, you can already see this comes closer to this distribution we have seen beforehand. So that means, first of all, Fisher postulated that this variation in phenotypic values results from additive genetic effects. And the more, so to say, genes we have, then the closer we come to this normal or Gaussian distribution. So that means now we have basically a nice explanation why we see this pattern, this kind of mixture of discrete and this continuous trait. This mixture means plants are maybe infected or non-infected, which is caused by single major genes, which cause this race-specific, this hypersensitive reactions, for example. And within this group, which does not carry this resistance gene, we see this continuous distribution because there are many, probably many small effect genes behind this distribution, yeah, making them more or less resistant, these plants. And yeah, for breeding, this has, of course, a very important impact. Say, in a classical plant breeding strategy, you can either go for these small effect genes, so to say, and yeah, behind them are quite often very general defense responses like lignin synthesis. That means just the plant builds a barrier against fungal infection, or kind of, for example, catinases, so to say the pathogen is attacked by enzymes. So all kind of nasty biochemical warfare is going on there. And this is really independent of the pathogen genotype, and so it's more durable but incomplete. On the other hand, this hypersensitive reaction that confers a complete resistance, so the farmer, for example, does not have to spray any fungicides if he wants to have a really clean field, but it's highly dependent on the pathogen genotype. And so quite often what breeders or also farmers have seen in the past, if a genotype was really resistant in the beginning and then its growth area increased over the years, then it also become more susceptible over the years because the pathogen population adapted. This led to this classical boom and bust cycles, where if we deploy, for example, one resistance gene and then it's in a lot of varieties, and then these varieties increase in area and it's very widely grown, and then this in parallel, the virulence gene of the pathogen also increases in frequency. And then at one point, so to say, all these plants are infected by the fungus, and we need a new resistance gene. Yeah? And this goes on and on and on. The problem is here, of course, that if breeder goes to the field, he can only see, okay, this plant is now completely resistant or a bit susceptible. But he does not necessarily know how many genes are behind this. And of course, let's see if we would combine multiple of these major resistance genes in one variety, maybe then would be more stable. We would kind of not see this boom and bust cycle. To do this, of course, we got to look into the genetic basis really behind this resistance. And it's basically the other branch we see here. It's not the quantitative genetics, but the molecular side of genetics. And of course, in the last 200 years, quite some progress was also made there, from stuff like that we know now, the structure of the DNA, and then in the 1980s or so, then we got molecular markers, and nowadays we have SNP arrays. So really, we can really look in the genome now. Basically what one would have done in the past, seeing this kind of gel electrophoresis, but nowadays we have even sequencing data, and we can, based with these chips or genotyping arrays, we can get thousands of molecular markers which cover all the genome in the plant. And one spin-off of this development is the integration of quantitative genetics and this molecular genetics, and also Mendelian genetics, in two approaches. This is genetic mapping and genomic prediction. So genetic mapping, or let's say in this case genomic association mapping, gives us some information. Where are the resistance genes in the genome and how many are there? Let's see, we have here, for example, the wheat genome, which has 21 chromosomes. And then, let's say we just look in chromosome 2A, and then we have a genetic map, and each of these small horizontal lines would now represent a marker we have on this chromosome. And based on a statistical test, one would just look, for example, at this marker, and then group our genotypes into two groups, which carry either the recessive or this dominant allele at this marker, and compare if they are significantly different, or if they differ significantly in their average, these two groups. So here, not the case, so probably this marker does not have anything to do with resistance. And then if we go on, okay, maybe there's already a small difference there, but not very strong, so not so super reliable. Just go on and go on, test each of these markers, and at one time, hopefully you will find a marker like this, where you can really see that if now plants carry this M3, large M3 allele homozygous, they are completely resistant. So then we know, okay, probably this marker now really tracks a resistance gene. And with this approach, you really can easily find, should say, the few big players that confer this complete resistance. And afterwards, one would try to combine these all these genes you have found in one variety. It's then called gene pyramiding. Let's say now, for example, we have these, in generation 1, these 3 parents, and each of them just carries one of these resistance genes. And the other, should say, in the favorable allele, the large R allele. Then we would try to combine all of these in one variety. So then we cross them, and then generation 2, okay, now we have progeny which already combines 2 of these resistance alleles, and then cross again, and then select the progeny which combines all of these 3 resistance genes in one variety. Of course, you can imagine how it looks very simple here, but there's a lot of effort behind it and many years of work until you get such a variety which really combines multiple resistance genes. But unfortunately, this has not been so super successful, especially in the long term, because the pathogen cracks one major resistance gene after another. So kind of we also need to target as many small players in the background, which confer only this partial resistance genes to have something like a second layer of resistance. And this can be done by genomic prediction. And I said with genomic prediction, you mostly target the many small and medium sized players in the background. And what we see just in the field is this kind of distribution, which is visible, so to say, but then invisible in this genetic background. It means you also find some genotypes which might be completely resistant in the foreground, but in the background, you can see, for example, if this resistance gene is overcome by the pathogen, you will see some variation. And of course, we would be more interested in, it's probably not visible, in finding then also the ones which are more resistant in the background genotypes. And here do these genotypes help us, which don't carry any of these major players. And with these, you can basically use these to assign an effect to each marker and link them to these minor resistance genes. Means quite often this is just based on regression models, where you then assign to each of these markers, and it's not even important to know where they are, an effect. Means some of these markers now are associated, let's say, with a resistance gene, which just switches or improves the resistance by 1%, or let's say even there's a medium-sized player there which increases the resistance by, let's say, 5%. And then you link these estimated effects with the genetic fingerprints of your selection candidates. And for example, this genotype 2, you can see here also then for this, let's say, this medium-sized player, it carries the favorable allele, so to say, no, the unfavorable allele, because yeah, the disease incidence would be increased, while genotype 1 carries the favorable allele there, which decreases the susceptibility or increases the resistance. And you just continue this exercise with all of these small effect markers. And then in the end, yeah, you sum across all these marker effects you have estimated for your genotypes, and you get a so-called genomic estimated breeding value. And genotype 1 comes out pretty well, just based on the prediction, would have a disease severity of 13%, so more or less resistant, or more on the resistant side, while this genotype 2 would have, in the background, you have, would be quite susceptible with 50% disease severity. Yeah? So that means one would then in the end find out, okay, which of my genotypes or selection candidates does not only carry major R gene, which is very nice for a farmer, yeah, to have complete resistant variety, but once such a resistance gene would be overcome, it's still not completely, yeah, so to say, yeah, eaten up by the pathogen. Yeah? And yeah, so to say, to sum this up, quite some work has been done in the integration of Mendelian, quantitative and molecular genetics knowledge, and it's also applied in modern plant breeding. And one example I tried to show here is this genomic selection of crop varieties with multiple layers of disease resistance. It means the first layer are these few major Mendelian genes, or these resistance genes that follow more or less a Mendelian inheritance. And then a second layer, these many small effect genes, these small and medium-sized players. And this has, of course, has the potential to prevent a catastrophic crop losses, yeah, when major resistance genes are overcome by evolving pathogen populations, and yeah, in general, reduce then also the application of plant protection agents like fungicides. And I would say this then one step towards a more sustainable agriculture. And with that, I'd like to thank you very much for your attention.
This video was supported by the project number ATCZ278 with the title Gregor Johann Mendel's Legacy to Science, Culture and Humanity, which is co-financed by the Interreg Austria and Czech Republic. Down below you see the title of the project in German and Czech. Here I would like to take a brief moment to explain what the project is about, because it is actually an awesome one. The main goal of the project is actually to promote Mendel's legacy by long-term cross-border collaboration in science and culture. There is a strategic plan developed that will promote this legacy in the fields of science and culture in Czech Republic, Austria, but also worldwide. This promotion will take various forms, be it meetings, workshops, publications, audio and video materials. One of the main parts of the project is actually rebuilding Mendel's original greenhouse in the Augustinian Abbey in Brno. This newly rebuilt greenhouse will be a new landmark, not just the city of Brno or the country of Czech Republic, but the entirety of the scientific fields of genetics as such. Mendel's greenhouse is the very place where genetics was born, with the first genetic experiment conducted on peas. Therefore, it is a really, really important location for the field of genetics. As a major outcome of this Interreg project, this greenhouse will become available for the general public, but also the scientific community as a multi-purpose gathering place for cultural and scientific events. This and much more you can find out if you visit the website gjm200.cz, which of course stands for Gregor Johann Mendel, and the 200 indicating the 200th birthday of Gregor Mendel, which we celebrate in 2022. Once again, a great thank you to the project and Interreg for the support of this video. Thank you for your time you spent on it watching. And from my side, I just wish you a very nice day.